Hi, good morning. My name is Mariana Quijada-Rivera. I'm a postgrad student of the Research Center of Food and Development from Hermosillo, Sonora, Mexico, and I'm going to present the work titled Analysis of Grapevine Zygotic Embryos Responses to a Cryopreservation Protocol by using Next-Generation Sequencing Technology.
Grapevine is one of the most important crops in the world. In 2018, the state of Sonora, Mexico was the leader in production. Until now, there is no an efficient method for the germplasm preservation of grapevine cultivars. However, in situ is the principal manner of grapevine preservation in this region, where the plants are vegetatively propagated in the field. In this condition, they are exposed to pathogen attacks and other biotic and abiotic stresses. Therefore, it's necessary to look for alternatives to preserve without changes the commercial and traditional valuable grapevine species. In this sense, an effective alternative for the germplasm preservation is cryopreservation, that can preserve for long term the plant genetic resources at ultra-low temperature, usually under liquid nitrogen. Different protocols of cryopreservation have been developed, and one of the most employed techniques is that based on vitrification. Vitrification can be defined as the transition of water directly from the liquid phase into amorphous phase, avoiding the formation of ice crystals by using cryoprotector components. However, several steps in the cryopreservation protocols can induce many biochemical responses in the tissues. The steps present in the protocol can cause physiological, molecular, and cellular damage. For those reasons, it is imperative the understanding of the complexity of the molecular mechanism involved, coupled with the optimization of the protocols to obtain high levels of regeneration plant tissues cryopreserved, and also to ensure fidelity of cryopreserved plants without phenotypic and molecular variations. About cryopreservation studies, it was found that the cryostage method is not being fully implemented within gene banks, even though when cryopreservation of Vitis vinifera tissue has been successfully reported. Most of the studies report are on the establishment of the optimal condition of cryopreservation in several Vitis vinifera tissues, measuring viability and percent of regeneration. But there are a limited number of publications investigating the effect of cryopreservation on molecular analysis involving gene expression. In this regard, Lazo-Javalera in 2016 reported variation of genome stability in cryopreserved buds. Genetic stability has been assessed in regenerants recovered from cryopreserved shoot tips by different protocols. Overall, the results obtained thus far are promising, not showing significant changes, even though the changes found were increases in polymorphic fragments due to exposing the tissue to PBS2 for a long time. This result provides some valuable information on controlling excessive exposure time to PBS in order to ensure genetic stability in regenerants following cryopreservation. Multiple publications studied the molecular effects of cryoprotectant reporting gene expression related to the oxidative damage and osmotic stress in cells. Most of the gene expression responses reported in tissues during cryopreservation are due to cryoprotectants or the whole process. That takes us to the needness of research and study genome transcriptome changes by freezing and thawing. We believe that this stage is one of the most critical steps for the regeneration of cryopreserved tissues. For this reason, the objective of this work is to identify by RNA-seq the genes differentially expressed in response to cryopreservation in zygotic embryos of grapevine. Our plant material sample collection of berries was obtained from Vitis vinifera, and the seeds were extracted. Grape seeds were incubated in gibberellic acid for 24 hours to break latency. After that treatment, the seeds were sterilized with the protocol reported by Lazo-Javalera in 2016. The zygotic embryos were obtained by seed dissection under a laminar flow hood using a stereo microscope. The cryopreservation procedure was made following the protocol reported by Garcia Coronado. The vitrification treatments were carried out using the plant vitrification solution number 2, PBS2, which consists of 30% of glycerol, 15% of ethylene glycol, 15% of dimethyl sulfoxide, and 0.4 molar of sucrose. The experiments were performed in 2 biological replicates with 100 embryos each one. The embryos were immersed in PBS2 at 50% and then in PBS2 at 100% at 4 Celsius degrees. After vitrification treatments, the embryos were stored in liquid nitrogen for 30 minutes and warmed for 3 minutes at 38 Celsius degrees. The PBS solution was eliminated and washed 3 times with sterilized distilled water. The samples were taken after vitrification solutions for treatment 1 and after the complete cryopreservation procedure for treatment 2. Total RNA was extracted from each of the biological replicates of zygotic embryos of treatment 1 and 2 using RNA-easy plant minikit. The RNA of each biological replicate was analyzed in the bioanalyzer chips and then used to construct the RNA-seq libraries. The libraries were sequenced in 1x75 single-length format on Illumina exec. For the gene expression and differential express analysis, the first steps was the evaluation of the reads quality using FastQC program, where we removed low-quality sequences. After that, we used Bowtie2 program for alignment of the high-quality RNA-seq reads with the reference genome of Vitis vinifera by Genoscope, using the default setting and with local alignment mode. We obtained the read count per gene using HTSEQ program. For the differential expression analysis, we use the software of Bioconductor Package HR. Finally, we use BLAST2Go program to estimate gene function. Read alignment to the reference genome result for the treatment 1 with PBS2 and its biological replicate, and for the treatment 2, where the embryos were exposed to PBS2 plus LN. We obtained an average of 96% of the overall mapped reach, with an average aligned concordantly exactly 1 time of 64%. More than 1 was 31% aligned with the reference genome of Vitis vinifera. The same values are a reflection of a good mapping, considering an average rate higher than 70% as acceptable. The analysis of the differences in Vitis transcriptome is important for the identification of the metabolic pathways induced during the cryopreservation process. First, to explore the correlation of the dataset, an MDS or multidimensional scaling analysis was done. The graph below shows how similar the samples of a treatment are to each other. It can be seen how the different treatments belong to different quadrants, which was favorable to continue with the analysis. It should be noted that normalized read per kilobase of transcript per million reads were used. The following graph is the volcano plot of the differentially expressed genes, which are those that show a significant historical difference in their level of expression because of the treatment, in this case, freezing thawing. Those genes with a change in their expression of more than 200 times are those with a full change logarithm greater than 1 and less than -1, and an FDR less than 0.05. which are the color dots, with pink being induced and purple being repressed. This analysis yields 229 differentially expressed genes, of which 186 are repressed and 43 are induced. Gene ontology enrichment analysis was performed to investigate overrepresented biological process or molecular functions involved in the genes that we found to be differentially expressed, obtaining for the induced ones the result of nucleosome assembly, nutrient reservoir activity, nucleosome, protein ubiquitination, etc. The pie chart shows the Go classification of the differentially expressed genes based on cellular component, molecular function, and biological process. On the other hand, the results with repressed genes show enriched process of methylation, calcium ion binding, cellular organizations, extracellular region, cytochrome C oxidase activity, ketene binding, etc. And the pie chart also shows the Go classification of the differentially expressed genes. Here are included the most induced and repressed genes, respectively. Some of the most induced genes were unnamed protein product, partial protein sphincter 2-like, heat shock protein, histones, cytochrome, etc. And some of the unrelated genes were receptor-like protein kinase, probable glutathione transferase, expansine, calmodulin, early nodulin, etc. After the whole protocol, most of the unregulated genes belong to plant cell wall integrity. Mechanical stress has been probably occurred during the freezing in liquid nitrogen. However, most of the upregulated genes were related with stress-responsive and nucleosome components. According to the data obtained in this study, the following hypothetical diagram shows the most differentially expressed genes and repressed genes. In the left part, the induced ones are observed, which coincide with the Go analysis. These genes are involved in processes related to the nucleosome and its modifications. Genes such as histones, in addition to heat shock proteins, are present as a defense in temperature changes. On the right side, the repressed genes are present, where membrane and cell wall components were mainly affected, like early nodulin, expansine, receptor-like protein kinase, etc., which indicates that cell damage are mainly in the membrane caused by freezing and thawing. This can be explained as cold stress can lead to a decrease in cell membrane fluidity and thus conformational changes in membrane proteins, accumulation of metabolites, and a change of the redox state in cells. Change in cell membrane fluidity and membrane protein conformation are the first physical change in the plant under low temperatures. In conclusion, we can say that the data obtained from the massive sequencing analysis can demonstrate that changes occur in the regulation of the genetic expression of 2029 genes due to frozen and thawing. The cryopreservation induced multiple metabolic responses in Vitis vinifera embryos, mainly the gene expression of several plant cell wall structures, secondary metabolisms, and some antioxidant enzymes. The transcriptome analysis shows enrichment in responses to stress and nucleosome modification process that attempt to maintain the integrity of the plant cell wall. These results bring us a deep knowledge of the holistic responses that freezing thawing causes in zygotic embryos tissues, and this will serve in the optimization of their cryopreservation protocols in the future. Thank you for your attention.